Rita Dobly, chairing Assessing GIS Estuarine Habitat Predictions with a New Statistical Approach for Genetic Signatures of Post-Glacial Recolonization.
Thank you. Thank you for that introduction, and thank you for coming out. I'll start off by introducing the three taxa that I'll be using for the work that I'll talk about today. The first two are species of goby fishes, which is the most specious family of fishes. It's Glyphes mirabilis and Cleotula licata. There are these two. The third is a species of killifish, which is Funculus parvipinus. And all three inhabit semi-tidal estuaries. So essentially, estuaries are just lagoons that have input of both fresh and marine water. And semi-tidal meaning that they're somewhat open to the ocean, so they have the tidal component. And the geographic expanse that I'll be talking about today for this study is from Southern California down to the middle of the Baja California Peninsula along the Pacific coast. So background, as has just been discussed, and as probably everybody knows, glacial and interglacial cycles have dictated climate for the last roughly million years, and as a consequence have also dictated sea levels. So these are glacial maxima, and this is present day. And so if we narrow in on flooding since the last glacial maximum, which was about here, we see a non-linear but significant change in sea level as we approach modern day limits around 7,000 years ago. So the difference is about between 120 and 140 meters of sea level increase over that time. Now, when you're working with estuaries, that's really important, right? So coast obligate species are going to change or move as the coastline moves over time. So this is just a little schematic sort of where we have, I've contoured depth by 10-meter bins from 0 meters, which is present day, down to 140 meters below sea level. So this is what the coastline, so this is the Baja California Peninsula. Los Angeles is right in there. This is what the coastline looks like today. This is what it looks like when sea level is 30 meters lower, 60 meters lower, 90 meters lower. This is what the peninsula looks like during low stand on average. And then you can see as the shoreline progresses back to where it is today. The other thing that's important to know about this region is that because of subduction of the Pacific plate beneath North America, the whole coastline tends to be very steep. There's an exception here and an exception here, but from here all the way down, there's really very little coastal shelf on which estuaries can actually form during this cyclicity. So there's two main objectives that I will be addressing today. The first is to estimate habitat availability from the last glacial maximum to today for these three species, and then the second is to essentially see if we can figure out signatures of post-glacial recolonization looking at refugial alleles specifically. So I'm more than happy to go through any of the methods in more detail with anybody afterwards, but this is going to be sort of a cursory overview for time's sake. So whether or not an estuary exists will depend on essentially two major physical factors. One is sea level, they occur at the interface of sea and land. The second one is slope. So we know that modern estuaries only form with very low where coastal slope is very low. So the way the habitat models are parameterized, the sea level curve I used was that of Chater et al. 2008, and that allows us essentially to go back and forth between what sea level was at the time and corresponding age. And then the second parameter is slope, and what I did was essentially we sampled for all three species over the geographical area of the study. We sampled all places that they're known to exist consistently year to year. And what I then did is in ArcGIS at all of those collection sites, I calculated the slope in at least 5 areas like this for each sampling location, and then used the maximum observed slope, which was 1.3%, as the slope cutoff value. So what I essentially said was, if modern estuaries don't form with a slope greater than 1.3%, then we can extrapolate that back and say that that's unlikely historically to have been able to support estuarine habitat as well. So the first set of results, I will show you, first of all, here are our collection sites with the letters corresponding to the species that was collected there. And then secondly, the depth is contoured from 0 to 140 meters below sea level, which corresponds with the last glacial maximum according to the sea level curve. And then I'll draw your attention to the insects. So 1 through 8 corresponds with the coastal location. red denotes areas that are between of the given sea level of this time period, so that's 120 to 140 meters or so below present sea level. Red denotes that the area was too steep to afford habitat at that time. Yellow indicates, like up here, indicates that habitat was perhaps possible during that time period. So what I will note is area 2, which is here, and area 8, which is here, and this is a big one, are the only sites along the coastline that are from this model thought to afford habitat during the last glacial maximum. So these are proposed refugial sites. Now switching gears and to the genetic side of things, what I did was develop, to complement mitochondrial data I had, I developed microsatellite data sets for the 3 taxa between 16 and 20 markers for each taxon. And what I did was actually use the modern day populations that are analogous to those 2 refugial sites, and I performed discriminant function analysis on those 2 populations, so essentially down here and then up here, that for those 2 populations within each species to find the alleles that segregate or characterize those 2 populations specifically to a significance level of 0.01 or better. And I will skip over some of these details, but what I did was then calculate the frequency of those alleles in all of the individuals at the intervening sites along here. So essentially we got this metric where put into a geographically explicit framework. 1 denotes the northernmost signature you can have, and 0 denotes the southernmost signature that you can have. There's one assumption and one caveat that I want to mention. Time is an important assumption in this case. We're not assuming mutation rates or anything like that. I know that that can be contentious to say the least, but one thing I will draw your attention to is, so we have a mitochondrial tree over here, 1800 base pairs that made with Mr. Vase. Then we have the microsatellite tree that I made using neighbor joining approach for the microsatellites. The difference, hopefully you can see, is the resolution. You have major polytomies here that are resolved with microsatellite data. These individuals are over that same geographic location, and there's 2 clades that are supported by a posterior probability of 1, but they're dated to about 630,000 years ago. The confidence intervals are in Ellingson 2012, I forgot to put them on. But the idea then is if you have greater increased resolution, partly because of the informational capacity of the data over the time period to differentiate between the more closely related types, that might, we're hoping, indicate, or the argument is indicate that these alleles are in fact evolving or reflective of processes that have occurred somewhat more recently than that. So that's the argument for age. The other sort of issue that we had to contend with was the fact that essentially, so these are the top 2 northernmost sites, Site 1 and Site 2. This is the North Conception Refuge that was identified previously. The issue is that this no longer supports habitat for today, and that is true for possibly 2 different reasons. One is, if we look at, based on the literature, there's every indication, I think, that this habitat formerly was semi-tidal, somewhat open, was a much bigger system. What you can see in historical maps, in addition to literature though, is this big system. So this is the map from the 1800s from the US GIS, and it is no longer so. So what you see, it's big, it's draining. The foothills, you can see today, it is actually, there's a sand bar that blocks it from actually connecting with the ocean, which means that you are essentially, so it's both dammed and levied at this point. It's dammed upstream, and the entire river here is levied for its whole course. And so what that means is you change the entire ecology of the estuary, and it supports entirely different species of estuarine fish than are found in the study. So the idea is it possibly has been extirpated more recently because of either human interactions or because of infilling in the last few thousand years from sediment offshore, and that is also found from the literature. So what we did, because we can't sample that population presently, is we used a population just north up here, and there's a few assumptions or there are a few ideas thinking why that might be okay. One is it always has a very northern signature in mitochondrial DNA. It's also the only northern site for all three taxa. So that was a caveat that I wanted to mention. So these are the results for the discriminant function analysis, where these are the three species. The x-axis, this is the northernmost site, and this is the southernmost site for each panel. And then this is the southern signature, and this is the northern signature. So obviously northern individuals have a more northern signature. That's great. This is using the discriminant refugio alleles. This is sort of a null, using the null alleles that are not significant. So you can see it looks like maybe we were able to partition the data in an informative way. But to zoom in on the top figure, these are delta AICC scores, and what you can see is we've done linear and cubic regressions among others that aren't shown, and there's actually the cubic regression for these two species based on the having a significantly better AICC score is preferred for the cubic regression than the linear regression. You can see this north-south break, or this north-south trend, which might be expected from a 2 n-member admixture model or isolation by distance. But there's some non-linearity component that is creating this curvature. So we do have north-south separation or trend, which makes sense, but there's some non-linearity component in at least 2 of these, if not this one also. And the current interpretation that we have right now is the fact that if we go back and look at our habitat reconstructions, what we're able to do is do it for every 10 meters of sea level increase, essentially. So what you see is actually habitat comes into creation or is originated at different times. So this is sort of an example of that. This is the purported North Conception Refuge, which has yellow inhabitable area for 20,000, 15,000, and 10,000 years ago. San Diego, however, does not come into origination until about 10,000 years ago, versus San Quintin, which is sort of the middle case where a lot of it does have habitat by about 15,000 years ago. The idea is if you can't distribute, if you only have a subsampling of habitats available, then that might create this nonlinearity sort of pattern that we observe. The other thing that I want to draw your attention to is, so this is habitat area and this is time, so this is last glacial maximum to now. You can see here you only have 2 different sites that are represented, which are the 2 proposed refuges. You have substantial increase in area of available habitat through time, and also all of these intervening sites come into existence from here to here. And then you have habitat peak at about 10,000 years and majorly drop off. This coincides with the modern still stand, where sea level reached about its current position, and this actually corresponds perfectly with anthropological and archaeological literature that says estuarine habitat of this kind peaked here and then fell off. So I'll conclude with future work. What I want to do is, I think the explanation makes sense, but I want to forward model this scenario to see if that the origination of habitat time can in fact correlate with this nonlinearity of the pattern observed, and extrapolate all the information I have. I have all the same data from Gulf of California as well. And not acknowledgements. Thank you very much for funding and thank you for